examples for you of what a data talk looks like, how to bring in a paper. This one is, I'm going to show you some rate equations. But these are rate equations for how an element gets in and out of cells, which is one of the main themes of the world from dust. So if you look at this, the kinetics of Pepto-Bismol is what we're going to talk to. So Pepto-Bismol, 3 guesses which element is in it. It is actually bismuth. And they found out if you made this bismuth salicylate, I believe that's what it is, that it's pink in part because the bismuth is pink in that form. But the thing is, if you have an upset stomach, you actually take the Pepto-Bismol, and one of the things it does is the bismuth kills bacteria. Okay, it's like an antibiotic, but the bismuth will also kill you. It's a heavy metal, and if you take too much, it will kill you. But the good news is you have a transport situation, so your cells can handle pretty high concentrations of it that bacteria can't handle. So you're basically, and it's completely safe for humans. Don't worry about taking Pepto-Bismol. I'm pretty sure that you'd have to drink gallons of it to have a bismuth overdose or things like that. And nobody wants to do that. So Pepto-Bismol will kill the bacteria because they don't have as good defenses against heavy metals. And this paper is this one right here with this kind of cool intro. By the way, a paper like this, you could talk about it as well. And be on the lookout for intro articles like this, commentaries, news articles that specifically talk about, even press releases sometimes you'll be able to find. Those will really help you understand what's going on because they're a different level of writing for a different audience, and they're really helpful. So if you see a commentary news article, press release, go for it, and definitely keep it together with the other thing. But this is the actual paper right there, and you see that it involves glutathione. Glutathione is actually mentioned in the world from dust, but I'm not sure how much I emphasize. Glutathione is related to a particular element. Anybody want to guess which element is in glutathione? What's that? Yeah, so you have the thiol, and you can even take it apart. It's actually got glutamine in it as well. I could ask which, now glutamine, gluten, those are a little bit confusing sometimes. But it's glutamine and sulfur. Or it might be glutamine, actually. I'm not sure which glue it is. But basically, glutathione has a part in detoxifying your cells from heavy metals. Because you have good glutathione and it can respond to the bismuth, Pepto-Bismol is no problem to you. Now, what's the reason for that? They basically looked at the entire system. So they did a lot of complex biology. They have this really cool figure. Whenever you see elements in a figure, at least I perk up. And I try to look at and see what they are. And bismuth is actually in the same group as arsenic and antimony. So this actually gets us back to Mono Lake. This talks about arsenic transport, which is similar to bismuth transport. And you can see that the blue is the bismuth transport, and so you have bismuth. You can put it into vesicles. You can export it from the cell. You need to keep it out of the cytoplasm in too high of a concentration. But you also have GSH, GSS. That's your glutathione because the sulfur is so important. The sulfur is good for binding the bismuth, and then you've got it grabbed on with the glutathione. Then you can put it into the vesicles, or you can export it. You can do whatever you want, but it is a detox mechanism that you have that works far better than what bacteria have. In fact, I'm not sure bacteria have it at all. And you see that actually glutathione is involved with these other colors. It's involved with copper response, gold response, and arsenic response. So there are 3 pathways that protect your cells from bismuth. The first pathway is that your cells just have a membrane that prevents the bismuth from getting in the first place. After they get in, you can attach it to glutathione and put it in a vesicle, or you can actually make more glutathione to sort of just attach and cover it up. So you can cover it up with glutathione, and or you can put it into a vesicle. So 1, 2, 3. And they talk about the whole study is which of these is the most important. Is it glutathione? Is it the vesicles? Or is it the membrane? And so they did the modeling, and they found out actually that number one may be the most important. The simplest answer may be the most important. And so they modeled how it gets into the cell. If you're modeling that, you're going to use rate equations. And you see actually right here, again, they use slightly different symbols than we use, but hopefully you can see the structure of these. You can see that there's an integrated rate equation right here. Here's time. Here's dj over dt. And if you look up here, what's J? J is the amount of solute in a cell at time t. So that's how much gets into the cell, and this is an exponential function. And then you see over here, J at time t, you basically integrate this, and you get a 1 minus e to the minus x term. This is exactly what we talked about, and this is high-level papers. So what I'm teaching you is not wrong. It's what they actually use in these papers, and it looks like it literally is Greek in some places. But if you can look through that to see the elements, you can see the rate law right here. This means rate versus time. And then you can see the e to the minus x, and you can say to yourself, hey, that looks like an integrated rate law. In fact, it's a first order integrated rate law, I believe. They don't say first order. I think they just sort of assume. A lot of times you just assume it's first order, just because it's unusual to be anything else. And so basically these equations, they look a little intimidating at first, but they actually are something that you can translate. You just have to look through to see what the symbols are that they're using. The basic structure of the math is universal. And I want you to be able to read something like this. So what you see right here is you have bismuth inside the cell over time, and they model, given passive diffusion, just the bismuth floating across the membrane. Even if it's the same type of membrane as far as bismuth getting across it, bacterial membranes, bacterial lipids versus human lipids, bismuth probably floats across those pretty much the same. There's probably not a specific bismuth defense. But the big difference between human and bacterial cells, remember, bacteria are the size of mitochondria, right? Human cells are much bigger. And so the size and the ratio of surface area to volume is what's really different here. And so when you have the bacterium, you have a much greater surface area to volume ratio. A lot less volume, a lot more surface area. Now, if you have the same, now you have to have like 1,000 bacteria that add up to one human cell. But you can imagine if each of them has their own membrane, that's a lot more membrane, a lot more exposed surface. And each of those is a surface that the bismuth can get across. The human cell, on the other hand, is a lot less membrane. And even if the membranes are no different to bismuth getting across them, there's a lot less chance for the bismuth to get across just because it's a bigger cell. And that's what they actually modeled this. They said, the rate constant for bismuth getting in is going to be dependent on the surface area to volume ratio. So if you have, is this why the bacteria are so much more sensitive to bismuth? They basically have a lot more surface area and a lot more chance for the bismuth to get into them. So we have all this fancy glutathione, and yeah, it helps. But the main reason why the Pepto-Bismol works is because this effect is actually the major effect. The fact that our cells are bigger. So it has a simple answer. At the end of the day, it has simple kinetics. You have an e to the x sort of function going up. This is from intracellular. So this is sort of like the product of the reaction of going from outside to in. You could talk about the concentration outside, but what they're interested in is the inside concentration. And you can talk about how fast this curves. This is going to be curving up differently depending on the rate constant. And the rate constant depends on surface area. More surface area, higher rate, more bismuth inside. So it's actually a kinetic effect. It's based on these rate equations, which are actually exactly what we showed you, believe it or not, just different symbols. So I think I'm going to go ahead and tell you, one of your test questions will be doing this. Like I give you this and I ask you some question about, can you identify which equations in your book these go along with? It's actually not that hard, and so I want you to be prepared to do that. You just have to have your book and be ready to find that chapter The faster you can find it, if you've done the homework and processed it in your mind, you should be able to do this one pretty quickly and gain time to spend more time on other problems. Or you can just not choose to do it and do the other problems. It's up to you. But I'm trying to give you an easy one. So, okay, so now it's your turn. And what I want to do is I'm going to give you a chance